Manufacturing requirements for ADC production plant. Facility design and infrastructure. Clean rooms. These are special rooms where the air quality, temperature, and humidity are strictly controlled. They are designed to reduce the amount of dust and microbes in the air to prevent contamination of the ADCs during production. Segregation. This means keeping different parts of the production process separate from each other. For example, the area where the cytotoxic drugs are handled would be kept separate from the area where the antibodies are produced. This is to prevent cross-contamination and to protect workers from exposure to hazardous substances. Waste management. This involves safely disposing of waste materials produced during the manufacturing process. Special procedures are needed for cytotoxic waste, which can be harmful to people and the environment. Equipment. Bioreactors. These are large vessels in which cells are grown to produce the antibodies used in ADCs. They provide the right conditions for the cells to grow and multiply. Chromatography systems. These are used to purify the antibodies and the final ADC product. They work by separating the different components based on their size, charge, or other properties. Conjugation equipment. This is used to attach the cytotoxic drug to the antibody. This is a delicate process that needs to be carefully controlled to ensure that the right amount of drug is attached to each antibody. Analytical equipment. This is used to test the quality of the ADCs. For example, HPLC systems can measure the purity of the ADCs, and mass spectrometers can determine their molecular weight. Personnel, qualified staff. These are people with the right training and experience to carry out the different steps in the ADC manufacturing process. This includes scientists, engineers, and technicians. Safety training. This is training provided to staff to ensure they know how to handle the cytotoxic drugs safely. This is important to protect the staff and to prevent contamination of the ADCs. Quality control and assurance. Testing laboratories. These are special labs where the ADCs are tested at different stages of the production process. This is to ensure that the ADCs are of high quality and are safe to use. Documentation. This involves keeping detailed records of everything that happens during the manufacturing process. This is important for tracking the production of each batch of ADCs and for showing that the manufacturing process complies with regulations. Regulatory compliance. GMP compliance. GMP stands for Good Manufacturing Practices. These are rules set by regulatory bodies like the FDA that manufacturers must follow to ensure their products are of high quality and are safe to use. Environmental compliance. This involves following rules set by environmental agencies for the safe disposal of waste, especially cytotoxic waste. Supply chain management, raw material sourcing. This involves finding reliable suppliers for the materials needed to make ADCs, such as the cells used to produce the antibodies, the cytotoxic drugs, and the linker molecules. Cold chain logistics. This involves storing and transporting the ADCs at low temperatures to keep them stable. This requires special refrigerated storage and transport facilities. Future considerations. Scalability. This means designing the facility so that it can increase its production if needed. For example, it might need to produce more ADCs if a new drug is approved for use. Adaptability. This means designing the facility so that it can change its processes or equipment as new technologies become available. This is important because the field of ADCs is rapidly evolving, with new methods and materials being developed all the time.